In this video, we're looking at BMAT 2019, Section 2, Question 1. So, in this question, we have a lovely diagram showing some steps from genetic engineering, and we're asked to work out what each of the letters correspond to. And we're given a long list here of things we can match up to the letters. So, let's start with the first one, W. We can see it's this little box here that's also present in this structure and this structure here. So we can see that it's been removed from this one here, which we should be able to work out as a chromosome just by the shape. So have a think, what makes up chromosomes and what could be removed from one? Well, we know that in fact genes make up the chromosomes and we can cut them out. So W is going to relate to the gene. So next we should look at X. X here is acting like a bit of a pair of scissors. It's going to snip out this gene from the chromosome, and it does a similar thing up here. It snips Y and opens it up. So what acts like a pair of scissors? What do we know that can cut open things? That is going to be a work of an enzyme, specifically a restriction enzyme. That's something that can cut DNA up. So X is our restriction enzyme. Next we have Y itself. So that's a circular thing at the top, and we can kind of guess if we're inserting some DNA into it, it's going to be a section of DNA itself. So what circular DNA do we know about? Well, that's going to be a plasmid, because plasmids are circular sections of DNA. So now we only have one thing left, Z, which is this down here. Now we can see that it's joining the DNA back into the plasmid, so it's sort of sticking it together. And what do we know that can stick things back together? That's going to be another enzyme, and the other enzyme we have on this list is the ligase. So ligases can patch the gene back into the plasmid, so our ligase is Z. So now we have all of our letters associated with the right terms. We know that our answer is going to be E.